On the MCAT, embryogenesis is tested as a strict chronological sequence. You have to track how a single cell systematically divides into a complex organism. This diagram shows fertilization. When a sperm meets the secondary oocyte, it releases acrosomal enzymes to penetrate the membrane. This contact triggers the cortical reaction, releasing calcium ions. This depolarizes the membrane, increasing the metabolic rate and creating an impenetrable barrier against other sperm. With the sperm's genetic material inside, the cell restores its normal diploid or 2N chromosome number. We now officially have a single-celled zygote. If two separate eggs are fertilized by two different sperm, you get dizygotic or fraternal twins. But if a single zygote physically splits into two separate masses, you get monozygotic or identical twins, who share the exact same genome. Looking at this illustration, we follow the zygote traveling toward the uterus. It undergoes rapid mitotic divisions called cleavage. One cell becomes 2, 4, 8, and 16. Notice the outer boundary remains the exact same size. The embryo's total volume isn't changing. By dividing into progressively smaller units within that fixed space, the cells increase their surface area to volume ratio. Lacking a formal life support system, the embryo relies entirely on this increased surface area ratio to exchange vital gas and nutrients relative to overall mass. Several divisions later, the embryo reaches the stage shown in this upper image. It's now a solid mass of cells called a morula, named after the Latin word for mulberry, which it closely resembles. Next, the solid morula undergoes blastulation. It hollows out to form the blastula, also called a blastocyst in mammals. As seen in the cross-section here, the blastocyst splits into two distinct groups, a fluid-filled cavity surrounded by outer trophoblast cells and a concentrated inner cell mass. The blastocyst eventually completes its journey through the fallopian tube and reaches the uterus, where it burrows directly into the endometrial lining to establish a permanent connection with the mother. This anatomical cross-section maps out what happens after implantation. Those outer trophoblast cells develop into the chorion, a membrane that eventually grows into the placenta to handle maternal-fetal gas and nutrient exchange. It takes time for the placenta to become fully functional. Until then, two other extra embryonic structures, the yolk sac and the allantois, visible near the umbilical cord, act as early bridges for fluid exchange and blood cell development. You can also see the amnion, the thin membrane tightly surrounding the fetus. It produces the amniotic fluid, which acts as a biological shock absorber, protecting the developing embryo from the physical impacts of maternal motion. The architectural split of the blastocyst sets up a division of labor. The outer cells are delegated to building maternal support systems like the placenta and membranes, allowing the inner cells to focus entirely on building the organism itself. Once implanted, that inner cell mass begins gastrulation. The bottom row of this graphic tracks how the hollow blastula invaginates, pushing its cells inward to generate a three-layered structure called the gastrula. These primary germ layers dictate the ultimate fate of every human body tissue. Ectoderm, or attractoderm, becomes features attracting us to others, skin, eyes, and the nervous system. Mesoderm, the mesoderm, forms the means to move physically and move fluids, the musculoskeletal and circulatory systems. Finally, endoderm forms epithelial linings like digestive and respiratory tracts. With the three layers established, the embryo initiates neurulation, the development of the nervous system. This phase is driven by the notochord, a small rod of mesodermal cells. The notochord induces the overlying ectoderm cells to slide toward each other, folding completely inward to create a deep neural groove. As those folds eventually meet and fuse, they pinch off to form a hollow structure called the neural tube. This tube is the direct foundation for the entire central nervous system. From a single fertilized cell undergoing cleavage, to a hollowed-out blastula tapping into maternal resources, to a three-layered gastrula sorting out the body's entire architecture. Mastering this chronological sequence and the specific organ fates of the germ layers is the key to conquering this section of the MCAT.